The brain is perhaps one of the most complex objects we know. Our brain is not only the seat of our intelligence, but also defines who we are, each of us as unique individuals. Because of its importance, it is supported by many physiological systems that protect its environment. The brain consumes energy at a great rate, taking 20% of the total energy we consume. That's more than 100 times the energy used in a smartphone, according to Google. Energy is not stored in the brain to any degree and so must be continuously provided via blood flow. And that brings us to the topic of this talk, the control of cerebral blood flow, often abbreviated CBF. A brief review of a complex subject. CBF must be controlled to cope with changes in arterial blood pressure. In this talk, it is termed autoregulation. Increased energy consumption due to neural activity, termed neurovascular coupling, and changes in the arterial partial pressures of oxygen and carbon dioxide. Here is an overview of the brain vasculature presented in the review by Willie and colleagues from the Ainsley Laboratory. You can see one of the major supply arteries, the carotid artery, with the carotid body at its bifurcation. Recall that the carotid body is the peripheral respiratory chemoreceptor that tastes the blood approaching the brain. It plays a role in the chemoreflex control of breathing, and is important for maintaining the acid-base status of the brain environment. Also pictured are some of the major cerebral arteries, like the middle cerebral artery. The main drainage vein is shown in blue, and after blood has passed through the brain, vascular resistance to flow, the high arterial pressure is lost to about zero. Before we turn to a consideration of the control of cerebral blood flow, a look at the anatomy of the cerebral blood circulation in greater detail. Here is a picture of the anatomy of the large vessels supplying the brain. On the left, the common carotid arteries arise from large intrathoracic arteries and give rise to the internal carotid arteries that enter the skull and merge into the circle of Willis. Vertebral arteries run along the cervical vertebrae, enter the skull, and join to form the basilar artery, which also merges into the circle of Willis. Picture B on the right shows a schematic diagram of the circle of Willis and its major branches. There are two important points to note. First, on the basis of pressure gradient measurements, extracranial arteries are responsible for about 20% of the total cerebrovascular pressure loss, an observation originally made by Faraci and Heisted in 1990. Thus, they are responsible for about 20 to 30% of the total cerebrovascular flow resistance. The other point is that the communicating arteries connecting the major supply arteries to make up the circle of Willis act as a compensatory blood flow distribution system in case of a supply failure in one of the major supply vessels, an anatomical flow protection arrangement. As shown here, Haczynski and Ostergaard proposed that the cerebral circulation should be considered as two different systems, a high-pressure system for the older, at least in evolutionary terms, part of the brain that deals with autonomic controls, and the cortical, low-pressure system with a more specialized control of flow distribution. Now recall that the central respiratory chemoreceptors are located in the older part, and so the control of blood flow may differ from that in the cortex. Indeed, observations from animal experiments suggest that blood flow in the region of the central chemoreceptors may be regulated to maintain flow in the face of an increase in arterial carbon dioxide, hypercapnia. Whereas in the cortical system, hypercapnia produces vasodilation and an increase in blood flow. If that happened in the region of the central chemoreceptors, it would wash out carbon dioxide and blunt the central chemoreceptor sensitivity to CO2. However, it should be kept in mind that blood flow control in the medulla has not been studied well, in contrast to that of the cortex. And from here on in this talk, it is the control of blood flow in the cortex that will be discussed. The distribution of cerebral blood flow and its overall adjustment to meet energy and oxygen demands and cope with changing conditions is accomplished by controlling the diameter of vessels so that the pressure loss along a length is increased. Simplistically, this control of diameter can be considered as changing the flow resistance, the ratio of pressure loss to blood flow over a length of vessel. As shown on the left, cortical blood flow is distributed bilaterally from the circle of Willis via major intracranial arteries, the middle cerebral arteries, MCA, the anterior cerebral arteries, ACA, and the posterior cerebral arteries, PCA. These feed their region of the cortex via penetrating arterioles to the capillary network supplying the tissues. On the right, this distribution system can be viewed using a hydraulic analogy as a flow resistance network, with resistance controlled by changing the vessel diameter via the smooth muscle lining vessels. Note the high resistance of the major supply arteries producing the 20 to 30% of the total pressure loss as mentioned earlier. When an increase in arterial PCO2 occurs, hypercapnia, and global blood flow increases, this high resistance produces a drop in overall cortical perfusion pressure. Consequently, blood flow in those regions unable to vasodilate sufficiently may decrease, in contrast to blood flow in regions with robust vasodilation, which will increase, appearing to steal the lost flow. This observation is known as the steal phenomenon. This figure 2 from Schaefer and Ayatokola's 2021 review shows the segmental heterogeneity of the cerebral arteries and the diversity of vascular and perivascular cells. Note that the vascular diameters indicated under the vascular segments refer to the human cerebral circulation. I draw your attention to several points. First, the internal carotid artery with its thick layer of smooth muscle cells surrounded by nerves arising from cranial autonomic ganglia. This is the extrinsic innervation embedded in perivascular connective tissue, the adventitia. The internal elastic lamina separates the smooth muscle cells from the endothelial cell monolayer. In the middle cerebral artery and pial arteriolar branches, the smooth muscle cell layer becomes progressively thinner, and a perivascular nerve plexus surrounds the vascular wall. Penetrating arterioles dive into the substance of the brain, surrounded by a perivascular space where macrophages and other cells reside. As the vessel becomes smaller, the intraparenchymal arterioles, the vascular basement membrane fuses with the glial basement membrane, and perivascular nerves are replaced by nerve terminals from interneurons or subcortical pathways. This is intrinsic innervation. In capillaries, smooth muscle cells are replaced by pericytes. More about these later. A final note is that the venous smooth muscle cells are morphologically, functionally, and molecularly distinct from arterial smooth muscle cells. This figure from Kistler et al. Illustrates the idea that cerebral blood flow is largely controlled by the parenchymal arterioles. The large pial arteries on the surface of the cortex contain multiple layers of vascular smooth muscle cells. Pial vessels branch into penetrating arterioles with only a single layer of smooth muscle cells, and then enter the cortical parenchyma, where microvessels are covered by pericytes. Do these pericytes control microregional blood flow? I quote the title of a paper by Hill and colleagues. Regional blood flow in the normal and ischemic brain is controlled by arterial or smooth muscle cell contractility and not by capillary pericytes. That was published in Neuron in 2015. More on this question in the next slides. Here is a detailed picture of a pial arteriole and penetrating arteriole. A pial arteriole on the cortical surface, giving rise to a penetrating arteriole, is shown on the right. Pial arterioles have a thick coat of smooth muscle cells, are surrounded by the subarachnoid space, and are densely innervated by nerve fibers originating from cranial, autonomic, and sensory ganglia, such as the sympathetic, parasympathetic, and trigeminal ganglia. And here is a more recent view of pericytes from Longden and colleagues in 2023, contradicting that of Hill and colleagues published in 2015. Capillary blood flow is regulated by electrical, as shown in figures A and B, calcium ions, as shown in figure C, and metabolic factors, as shown in figure D, all of which involve pericytes. And image E presents a top-down view of an arteriole enwrapped by smooth muscle cells and lined by endothelial cells diving into the brain. On the proximal capillaries close to the arteriole are a population of pericytes that are contractile, that express a high alpha smooth muscle actin, and rapidly regulate underlying vessel diameter. Deeper capillaries are covered by the cell bodies and processes of thin strand pericytes that express a low alpha actin and regulate diameter over longer time frames and more modestly. And in this quite complex figure here, which you can study at leisure, here is more experimental evidence from the mouse cerebrovasculature to qualify the parasitic control of blood flow in that it is substantial but slow. And finally, here is the most recent view of pericyte blood flow control versus smooth muscle control of cerebral blood flow by Davis and Atwell, online publishing in 2023. And they make several points as shown here. First, both pericytes and smooth muscle cells are major contributors to blood flow control. Second, since capillaries have a smaller diameter than arterioles, this increases the effect of pericyte contraction on vascular resistance. Third, capillaries dilate before arterioles, providing the first reduction in vascular resistance. And finally, pericyte-mediated capillary constriction increases the probability of capillary occlusion by neutrophils and other blood cells. This picture summarizes the elements controlling the cerebral blood flow, and each segment of the cerebrovascular tree is characterized by diverse vascular and perivascular cells. The vascular and astroglial membranes delimit the perivascular space, which disappears when these membranes fuse together. Pial arterioles give rise to penetrating arterioles, the first order branch of which is defined as precapillary arterioles. For mural and endothelial cells, the genes enriched in each vascular segment are also indicated in this diagram. Maintenance of cerebral blood flow in the face of changes in arterial blood pressure is termed autoregulation. However, this term is often used indiscriminately to describe any type of blood flow regulation, so be careful. Smooth muscle cells detect intravascular pressure changes via mechanotransductive cytoskeletal proteins. Integrin links extracellular matrix proteins, such as collagen, to actin fibers that permit extracellular mechanical signals to be directly transmitted into the cell. Actin responds to this mechanical input by rapidly changing the ratio of F-actin to G-actin, and also acts as an intracellular sensor. Cadherin junctions mechanically link adjacent cells, and gap junctions link cells electrotonically. And in this way, the population of smooth muscle cells is coordinated to act as a unit. This graph from Knott and Nelson shows the variation of membrane potential and calcium concentration with intravascular pressure. The resting membrane potential of smooth muscle is approximately -50 to -40 millivolts. In arterial smooth muscle, this membrane potential is sufficient to open calcium channels, elevate global intracellular calcium concentration, and cause a tonic constriction. Hyperpolarization to -60 millivolts lowers calcium concentration to about 100 nanomoles. And depolarization to about -30 millivolts elevates calcium concentration to about 300 to 400 nanomoles. These changes in global intracellular calcium concentration are sufficient to cause maximal dilation and constriction, respectively. Arterial blood pressure is pulsatile, and consequently autoregulation can be considered to operate at all its component frequencies, which would be observed in a Fourier analysis spectrum. Simplistically, however, autoregulation function is subdivided into two categories, static and dynamic. Static, the low-frequency range autoregulation, controls the vessel resistance so as to maintain a constant cerebral blood flow despite variations in the mean arterial blood pressure. Lassen's classic static autoregulation curve is shown on both left graphs, and it describes the constancy of cerebral blood flow over a range of between 50 and 100 millimeters of mercury arterial blood pressure. However, the actual regulation range is much less, as shown in the lower right graph, which is based upon more recent observations from the Ainsley laboratory. Dynamic or high-frequency autoregulation controls the vessel resistance in response to pulsatile blood pressure. Transfer function analysis, sometimes abbreviated TFA, is often used to assess the autoregulation of cerebral blood flow at the frequencies of pulsatile blood flow in terms of gain and the change in the cerebral blood flow per change in blood pressure, and the phase relationship or lag of the cerebral blood flow changes in response to these blood pressure changes. And these measures can be used as an assessment of the health of dynamic autoregulation. Here is a more detailed examination of autoregulation over a range of frequencies by TAN that shows how the range at which cerebral blood flow is regulated decreases as the blood pressure fluctuation frequency increases. These observations suggest that the mechanisms involved in autocorrelation are likely the same for all frequency, and the separation into static and dynamic aspects may therefore be misleading with regard to the physiology involved. Although this talk has separated the control of cerebral blood flow into three major aspects, it must be kept in mind that all three operate to control blood flow via a common pathway, the cerebrovascular smooth muscles. Consequently, there is competition for control, and each regulator can interfere with the other. This illustration from Willie and colleagues' review show that CO2's direct effect on the smooth muscle to change cerebral blood flow with CO2 declines as the arterial blood pressure decreases. When neural activity in a region increases, energy and oxygen usage also increase and must be supplied by an increase in the regional blood flow. What mechanisms link the increased neural activity to the increased blood flow? The neurovascular control of cerebral blood flow is referred to as neurovascular coupling, sometimes abbreviated NVC. On the left, a figure illustrating the energy supply, usage, and blood flow regulation in the brain. Parts B and C describe the ways by which neural activity could affect cerebral blood flow, either through a feedback control system to match energy supplied to energy demanded, or a direct effect of neural activity on cerebral blood flow. It is the latter feed-forward system that is currently thought to be the mechanism of neurovascular coupling. However, experiments by Hosford and colleagues in 2022 found evidence for the feedback aspect, with CO2 acting as the signaling molecule. On the right, this figure illustrates one of the major theories of neurovascular coupling, showing both local and remote vascular components. Excitatory neurons have a powerful effect on local neural activity and energy metabolism, and may drive local vascular changes through neurotransmitters, vasoactive ions, as well as the byproducts of neural activity. Astrocytes are also thought to participate in this process, as well as interneurons that may signal blood vessels through direct neurovascular connections, releasing neuropeptides and nitrous oxide. This rather complicated figure presents evidence showing that astrocytes are sensitive to physiological decreases in the partial pressure of oxygen. Panel A presents in vivo imaging of hypoxia-evoked astrocytic calcium concentration responses in the somatosensory cortex of an anesthetized adult rat. At the top, pseudo-colored images showing changes in the fluorescence taken at the times indicated by arrows on the bottom panel, and at the bottom, traces from cells showing changes in astrocytic calcium ion concentration in response to hypoxia, with traces 5 to 7 showing non-responding cells that were not labeled. The inset shows average changes in the fluorescence induced by hypoxia in 10 labeled cells and 5 neighboring cortical cells that were not labeled, recorded in the experiment. Panel B displays hypoxia-induced calcium concentration changes of carotid body glomus cells in culture, visualized using the calcium indicator FURA2. And inset are pseudo-colored images of a cluster of glomus cells showing changes in the fluorescence in response to hypoxia. The PO2 threshold of activation was 40 millimeters of mercury. Panel C displays oxygen responses of brainstem slice astrocytes with a hypoxia-induced calcium concentration threshold of activation of 15 millimeters of mercury. Finally, panel D shows a simultaneous imaging of hypoxia-induced changes in the indicator and calcium concentration in cultured brainstem astrocytes, showing that mitochondrial depolarization precedes the calcium responses. These authors suggest that the hypoxia sensor of astrocytes resides in the mitochondria, in which oxygen is consumed. Hypoxia inhibits mitochondrial respiration, leading to calcium release and vesicle release of ATP. And I note that this is a fail-safe mechanism. This picture shows how brain-intrinsic vasoactive signals evoked by neurovascular coupling and possibly central pathways arise within the substance of the brain and propagate retrogradely to microvascular smooth muscle cells and beyond via interendothelial junctions. This is a retrograde vasodilation effect. The local vascular response elicited by overall neural activity leads to hyperpolarization of endothelial cells through potassium channels. Whether mesh and thin strand capillary parasites participate in this process remains unclear at the moment. Hyperpolarization is propagated retrogradely through interendothelial gap junctions and is transmitted to contractile mural cells, actin-containing and sheathing parasites, and smooth muscle cells, likely via myoendothelial junctions and potassium channels. In turn, mural cell hyperpolarization suppresses voltage-gated calcium channel activity, resulting in intracellular calcium depletion, relaxation of the contractile apparatus, and vasodilation. The relaxation is then transmitted to adjacent mural cells through intercellular gap junctions, leading to retrograde vasodilation, which eventually reaches pial arterioles at the brain surface. So, how is vascular smooth muscle tone controlled by the endothelium? This schematic diagram shows three major mechanisms of smooth muscle vasodilation from the endothelium. They involve activation of adenylate cyclase, potassium channels, and guanylate cyclase. Both endothelium-dependent and endothelium-independent mechanisms can produce vasodilation. For example, several peptides, beta-adrenergic agonists, and vasodilators such as nitroglycerin act directly on vascular smooth muscle to produce relaxation. Carbon dioxide is also a potent vasodilator, but will be discussed later. Prostacyclin, which is released from endothelium, beta-adrenergic agonists, and several peptides activate adenylate cyclase and thereby produce vasodilation. And vasodilation in response to EDRF, the so-called endothelial-derived relaxing factor, which is nitric oxide, is mediated by activation of guanylate cyclase. In addition, potassium channel openers increase the open probability of potassium channels, hyperpolarize vascular smooth muscle, and thereby relax blood vessels. This figure depicts experiments showing that NO is one of the major transmitters involved in transferring the signals carried by endothelial cells to arterial smooth muscle cells. As shown in these experiments, blocking nitric oxide transmission blunts the neurovascular coupling increase in blood flow. And here is more evidence about the involvement of NO, where nitric oxide inhibition in healthy adults reduces the resting regional and total perfusion. In 1998, Faraci and Heistad proposed that regional vasodilation increases blood flow in one region, which results in an increase in flow in the upstream large arteries. And these large upstream arteries then experience a flow-mediated vasodilation. What is flow-mediated vasodilation and how does it work? Now, according to Wikipedia, flow-mediated dilation, abbreviated FMD, refers to the dilation of an artery when blood flow increases in that artery. The primary activator of FMD is thought to be shear stress, sensed by endothelial cells which release nitric oxide to relax smooth muscle cells. Measuring FMD is used as a non-invasive assay to detect endothelial dysfunction. And here is a figure from a review by the Ainsley Laboratory showing the various transmitters and their roles hypothesized to control vascular smooth muscles. I draw your attention to the top of the diagram to note that shear stress plays a role and is generally accepted as the mechanism for flow-mediated vasodilation. What is shear stress and how does it relate to flow-mediated vasodilation? In the next 3 slides, I take a diversion to explore some personal ideas about flow-mediated vasodilation that are contrary to current understanding. This figure illustrates the forces involved in blood flow through a vessel. It is easy to see that circumferential stress results from the internal pulsatile pressure. In response to this cyclic circumferential strain, endothelial cells respond morphologically with alignment of the cells perpendicular to the force vector, subsequently followed by phenotypic changes. The mechanism by which the endothelium recognizes and transduces mechanical stimuli involves various unspecified signaling systems. Shear stress, however, is not so easily envisioned and involves viscosity and the geometry of the flow. I draw your attention to the parabolic flow profile, showing that the blood velocity next to the endothelium is much lower than in the center of the vessel. As a result, and as these authors note, shear stress, in contrast with circumferential stress, is a relatively weak force. Therefore, highly sensitive mechanisms seem necessary to sense shear stress, including the detection of complex patterns of shear. Is such a weak link important physiologically? In this experiment from the Ainsley Laboratory, a brief increase in carbon dioxide produces a delayed transient vasodilation of the internal carotid artery. The increase in the internal carotid artery diameter occurs following both the return of carbon dioxide and the calculated shear stress to baseline levels. Shear stress here is measured as the area under the curve of a calculation based upon Doppler ultrasound measurements of a combination of velocity and diameter. In a circular tube, for example, with a Newtonian fluid, the shear stress is the product of viscosity and 4 times the mean blood velocity divided by the vessel diameter. In these experiments, the shear stress area under the curve, AUC, was positively correlated to the magnitude of vasodilation. That followed removal of a statistical outlier, which is circled, and that it depends upon nitrous oxide, we know. The authors suggest that this evidence implicates shear stress, as a potential regulator of cerebral conduit artery vasomotor tone. In further experiments from the Ainslie laboratory, I note they found that nitrous oxide is involved in the ICA diameter experiments using a transient CO2 stimulus, but not for CVF experiments using steady-state CO2 changes. And this observation led to my alternate explanation for the CO2 shear stress transient ICA responses. I suggest that CO2 vasodilation of the intracranial arterioles feeds back retrogradely via the endothelium, as shown in the boxes 1, 2, and 3. Here's the scenario. CO2 produces an immediate smooth muscle vasodilation of cortical arterioles downstream from the supply arteries, increasing cerebral blood flow in the large supply arteries. The downstream sudden change in circumferential stress is sensed by the endothelial cell lining. Mechanotransduction promotes a signal traveling retrograde via gap junctions. The cortical epithelial cells thus relay the stimulus upstream with release of NO in the large conduit arteries to cause vasodilation. I note that this relayed signal is brief, due only to the sudden change in circumferential stretch. So this scenario would explain the delayed major vessel dilation and the involvement of NO. It also means that the stretch signal is proportional to the rate of change and therefore transient. So the transient response depends on NO, but the steady-state response does not. It suggests different mechanisms at work here. Here are examples of middle cerebral artery percent changes in transcranial Doppler velocity as they respond to a step increase in end-tidal PCO2 of 10 millimeters of mercury. For a step of normocapnia to hypercapnia, subjects 1 and 2 showed a short time constant exponential response. Subject 12 showed an initial fast response that faded and was replaced by a slow response. And subject 22 showed an oscillatory response. For a step from hypocapnia to hypercapnia, straddling the resting end-tidal PCO2, subjects 24 and 25 both showed an overshoot in the initial response. So these observations raise the question, do the oscillations like these indicate a competition for control of vascular smooth muscle between various mechanisms? Here are some last words on the subject of flow-mediated vasodilation and its relation to shear stress, and a general conclusion concerning the role of the epithelial cells lining the cerebral blood vessels. I suggest that shear stress is too weakly produced to be an effective stimulus. Circumferential stress is more robust and produces the appropriate signals for the flow-mediated vasodilation effect. Regardless of the mechanism involved, the cerebrovascular epithelium acts as a network to coordinate smooth muscle control, spreading a regional vasodilation effect from, for example, neurovascular coupling to other parts of the vascular tree. Epithelial health is therefore of vital importance. We now come to the last of the three factors affecting cerebral blood flow, changes in the arterial blood gases, carbon dioxide and oxygen. Despite the efforts of the respiratory chemoreflex control system, various circumstances can disturb this regulation of arterial partial pressures of oxygen and carbon dioxide. How does cerebral blood flow respond? Here is a pictorial overview of the vasodilatory effect of carbon dioxide, showing data assembled from various sources and experimental techniques in a review from the Ainsley Laboratory. These observations at various points in the vascular tree show that vessel diameter increases at all levels of the cerebral vasculature. In A, the precapillary and capillaries. In B, a Peel vessel. In C, the middle cerebral artery, and in D, the internal carotid artery. Cerebral blood flow changes with blood gases, oxygen and carbon dioxide, and this graph shows transcranial Doppler, TCD, measurements of middle cerebral artery blood flow velocity. As the arterial partial pressure of CO2 increases from 25 millimeters of mercury, hypocapnic, to 55 millimeters of mercury, hypercapnic, at isoxic tensions of both 50 millimeters of mercury, hypoxic, and 150 millimeters of mercury, hyperoxic, the middle cerebral artery velocity increases in a sigmoidal fashion. Comparing the solid and dotted line fits, it can be seen that hypoxia enhances the response to CO2. The sigmoid fit to these data results from the vascular limitations of vasoconstriction in hypocapnia and vasodilation in hypercapnia. Note, too, that blood pressure, the blue lines, was also measured and acts as a confounder because changes in blood pressure also affect blood flow directly. In this individual, blood pressure was constant until a threshold PCO2 was reached around 45 millimeters of mercury. Above, blood pressure and cerebral blood flow increased linearly with PCO2. The constant blood pressure below the threshold enabled the sigmoid vasodilation response to CO2 to be seen unaffected by blood pressure changes. Above the threshold, however, the increasing blood pressure increased cerebral blood flow proportionately. The picture at the right is a brief overview of how CO2 changes vessel diameter and the resistance to flow. CO2 disrupts the regulation of intracellular hydrogen ion concentration in smooth muscle cells and changes the tone via mechanisms involving changes in calcium and potassium ion concentrations. More on this later. These figures show the effect of a decrease in the oxygen supply to the brain, whether by hypoxia or anemia. The left graphs show the interaction of hypoxia with the vasodilatory effect of CO2. The same middle cerebral artery flow velocity data are plotted against PO2 in the upper graph and against PCO2 in the lower graph. The response to hypoxia is nonlinear, with substantial change only evident below a PO2 of 55 millimeters of mercury. On the right, the graph shows the effect of anemia on cerebral blood flow, a quite different relationship between cerebral blood flow and the hemoglobin concentration. As will be seen, blood flow in both these circumstances is regulated so as to provide the oxygen supply demanded. How is vascular smooth muscle tone controlled? In the next slides, I develop a model scenario that describes a hypothetical mechanism. The experimental observations shown here demonstrate that there is a direct relationship between intracellular hydrogen ion concentration and smooth muscle tone. Changing extracellular pH alters intracellular pH and smooth muscle tension in these experiments. This figure illustrates results from experiments showing the linkage between pH changes and calcium ion concentration, of which the latter is the regulator of smooth muscle tone. The authors note that nanomolar changes in intracellular hydrogen ion concentration lead to nanomolar changes in intracellular calcium ion concentration. And the authors postulate a coupled exchange ion transporter for calcium and hydrogen ions. Viewed from the understanding of the Stewart approach to acid-base, I suggest that hydrogen ion is not transported across membranes. Intracellular hydrogen ion concentration is the result of carbon dioxide levels and the intracellular concentrations of these strongly dissociated ions. Here are illustrations from 2 articles emphasizing the intracellular regulation of hydrogen ion concentration. They show that this regulation involves the movement of strongly dissociated ions like sodium and chloride across the smooth muscle cell membrane. Note the bicarbonate-centric approach also used by these authors. In the Stewart approach, hydrogen and bicarbonate ions are not transported across membranes, but determined by the PCO2 and the difference between the concentrations of positive and negative strongly dissociated ions. The strong ion difference, SID. This figure summarizes some of the complex interactions that determine the control of smooth muscle tone, and also includes the interaction between endothelial cells and smooth muscle cells. Details of the linkage between CO2 and tone via changes in hydrogen ion concentration regulation remain beyond a simple description suitable for this talk, and can be sought in the various publications referenced. Again, I suggest that although these authors show a bicarbonate-centric approach to acid-base regulation in the smooth muscle intracellular space, the regulation of ion movements across membranes can easily be viewed from the Stewart point of view. Here is my model of a cerebrovascular smooth muscle cell, showing the factors involved in the control of intracellular hydrogen ion concentration, which, via changes in calcium ion concentration, determine smooth muscle tone. As the Stewart approach describes, hydrogen ion concentration is a function of PCO2 and strong ion difference, SID. Regulation of hydrogen ion concentration therefore involves the movement of strong ions, chloride, sodium, and potassium across the cellular membrane. Note that such membrane transfers require energy and oxygen. More on this aspect later. These ideas can be assembled into a conceptual model control system that can be simulated mathematically. And here is a block diagram of such a system. Deviations from normal resting hydrogen ion concentration operate to transfer ions, altering the strong ion difference, SID, to restore hydrogen ion concentration internally to normal. Carbon dioxide changes hydrogen ion concentration and thus acts as a disturbance to this system. In addition, energy and oxygen are required by the SID controller, and so changes in arterial oxygen concentration also act as a disturbance to the system and affect its function. Comparing the model CBF response to CO2 pictured in A with an observed response in B shows the model is capable of simulating actual observations with respect to the CBF response to CO2. Nevertheless, as in all modeling, the caveat is that this correspondence between simulated and actual observations does not necessarily mean that the model is correct, just that it is not wrong, and the hypothesis is therefore feasible. Turning to the way the cerebral blood flow responds to changes in the oxygen supply, I suggest that the control of cerebral blood flow is fail-safe with respect to oxygen supply. A fail-safe system is designed so that when a failure occurs, it causes an action to mitigate that failure. One of the best-known fail-safe systems is train control, where the driver must hold a switch closed so that if the driver fails to do so, the train stops. Fail-safe systems in physiology can operate to either permit or prevent a process. And here illustrated are two valves that require air pressure to either keep open or closed. The valve on the left requires air pressure to keep it closed. When the air pressure fails, the valve opens to allow flow. And the valve on the right requires air pressure to keep it open. When the air pressure fails, the valve closes to prevent flow. The idea of a fail-safe view of oxygen supply comes from viewing the way that HIF-1 alpha works. HIF-1 alpha control is a fail-open valve system, since failure to supply oxygen leads to failure to destroy HIF-1 alpha, which then activates numerous systems to mitigate the loss of oxygen supply. The fail-safe scenario for cerebral blood flow control and oxygen supply can be incorporated into the previously described model of cerebral blood flow control by CO2. Cerebral blood flow control, like that for HIF-1 alpha, is also fail-open, since failure to supply oxygen leads to a relaxation of arterial or smooth muscle to increase blood flow and mitigate the loss of oxygen supply. The regulation of strong ion difference SID in the model to control the intracellular hydrogen ion concentration requires a supply of oxygen. As the supply of oxygen, DO2, decreases, the SID controller fails, and hydrogen ion concentration cannot be maintained constant and increases. Consequently, via decreases in intracellular calcium ion concentration, smooth muscles tone cannot be maintained. Vessel diameter increases and CBF increases, restoring the oxygen supply. These graphs comparing CBF responses of the model, the black lines, to experimental data, the red dots, in A from hypoxia and in B in anemia, demonstrate that the model is capable of simulating the observations. So, the idea of the fail-safe supply of oxygen and cerebral blood flow is not wrong, and the fail-safe hypothesis is feasible. The model that I have presented provides a possible mechanistic view of the control of cerebral blood flow during changes in arterial blood gases. However, while the model links changes in oxygen supply and smooth muscle hydrogen ion concentration to vasodilation, it does not specify details of the links between hydrogen ion concentration and vascular tone. Now this figure shows the movements of ions calcium, chloride, and potassium across the smooth muscle cell membrane to produce vasodilation and vasoconstriction, and it illustrates the complexity of the controlling factors. Recall that experiments from the Ainsley laboratory show that NO is not involved in the cerebral blood flow response to CO2. We must therefore look for a link between intracellular hydrogen ion concentration, membrane potential, and calcium concentration. And a likely candidate is the potassium channels. We return to the question of how vascular smooth muscle tone is controlled in this slide. As this diagram illustrates, potassium channels appear to be a key controller of membrane potential in both endothelial cells and smooth muscle cells. In both, intracellular potassium concentration is high, so that opening potassium channels allows positive charges to escape and causes a hyperpolarization of the cell. Because endothelial cells do not express voltage-sensitive calcium channels, this hyperpolarization of the endothelium enhances calcium influx through receptor-operated calcium channels. And this increase in cytoplasmic calcium concentration leads to production of endothelial-derived relaxing factor, EDRF, or packed nitrous oxide, and or prostacyclin, PGI2, from the endothelium. Now, in contrast to endothelial cells, hyperpolarization of smooth muscle cells reduces the open probability of voltage-sensitive calcium channels and decreases cytoplasmic calcium concentration of vascular smooth muscle. And this decrease in the cytoplasmic calcium concentration reduces the calcium-dependent contraction of vascular smooth muscle to produce vasodilation. Here are further illustrations of the mechanistic linkage between potassium channel opening and vascular tone. The diagram on the left shows how potassium channel opening changes intracellular calcium via changes in membrane potential, and the diagram on the right provides a graph of this relationship between membrane potential and intracellular calcium ion concentration. It therefore seems logical that CO2 might act via changes in intracellular hydrogen ion concentration to open potassium channels, as shown in this diagram. However, it is important to remember that intracellular hydrogen ion concentration is regulated by changing the concentration of strongly dissociated ions like potassium to alter the strong ion difference, or SID. CO2 increases intracellular hydrogen ion concentration, and any increase in SID to correct this acidosis due to the CO2 would act to depolarize. But in both endothelial and smooth muscle cells, this action opposes the hyperpolarization and vasodilation that occurs with CO2. So it must be concluded that CO2 overwhelms this regulation of intracellular hydrogen ion concentration, as my previous model presented hypothesized. While such regulation may operate, there is always an error, and CO2 will increase intracellular hydrogen ion concentration. It is this increase in hydrogen ion concentration that may affect potassium channels, and relaxation follows, as this diagram describes. However, the link between hydrogen ion concentration and opening potassium channels is not firmly established experimentally. And it should also be noted for this scheme that astrocytes are sensitive to CO2, releasing factors affecting both endothelial and smooth muscle cells. I conclude this talk with some final words. 3 control systems act to protect the supply of cerebral blood flow. Blood pressure autoregulation to cope with systemic blood pressure changes. Neurovascular coupling to cope with increases in regional demands for increased flow. Respiratory chemoreflexes to maintain arterial blood gas tensions of oxygen and carbon dioxide. These systems involve alterations in the flow resistance of cerebral blood vessels via changes in vessel diameter throughout the cerebrovascular tree, from the major supply vessels to the capillaries. The majority of control is exerted by vascular smooth muscles, which act as a final common pathway actuator. Cerebrovascular sensors involved include mechanotransduction in both vascular smooth muscle and in epithelial cells, with the latter communicating with smooth muscles. Gap junctions connect epithelial cells into a coordinating network, with transmitters such as nitrous oxide communicating with muscle cells. Blood gas disturbances also act directly via changes in the smooth muscle intracellular control of hydrogen ion concentration.